Hello and welcome to our lecture on Chapter 8. In this chapter we're going to be taking a basic look at some of the elements of infectious disease. And we're going to do this so that we can have a discussion that specifically addresses emergent diseases. So the first thing we want to know is, what is a pathogen? So pathogens are microbes that can cause disease. And we talked about these in our previous chapter, recognizing that we're talking about things like parasitic worms, various protozoans that can act as disease-causing agents, even fungi. But specifically in this chapter, we're going to take a look at bacteria, which are a prokaryote, that is, an organism without a nucleus, and two non-living things that can cause disease, viruses and prions. Okay, so we want to have some general idea about how things are spread. And I just included this graphic here to give us an idea that some things are spread by a vector, say a mosquito. Some things may come through our food or maybe are transmitted on some kind of object. We have direct contact, and we might even include droplets here which spread only a very short distance as being something close to direct contact, as opposed to, say, airborne transmission where something can stay in the air for longer. And these are going to relate to how easy it is for a particular disease to spread. Now we're going to start with bacteria, and again, our number one idea here is that these are prokaryotic organisms. We know that they're going to be much smaller than human cells, about 1/10 the size, but they may be 4 times larger than viruses, these non-living particles we'll talk about next. So these guys are going to have a single chromosome, that's the nucleoid we see over here. Although we do have plasmids which are also going to contain DNA and potentially carry interesting things that could be transmitted from one bacterium to another. Something we need to know about bacteria is that in addition to a plasma membrane like we have around our cells, they also have a cell wall and a capsule that surrounds that. The cell wall is made of peptidoglycan. It's a combination of protein and carbohydrate, and because we don't have anything like this, we can make certain drugs, the psyllin antibiotics for example, that can target this peptidoglycan or target the cell wall, meaning that we can cause destruction of bacteria utilizing these. Now this outer coating is interesting in that because it's slimy or has other things that allow the bacterium to stick to something, we can create what we call biofilms as a result of these. The one you're probably most familiar with is that kind of sticky, slimy feeling you get on your teeth after you've gone a while without brushing them. But you might also take a look inside the pipes that are draining your sink to find other biofilms that we find around. Now, of course, these things can even serve as a defense against white blood cells. So we have not only a plasma membrane, but a couple of other layers that are around a bacterium that we might see as targets or which might work as defense of the bacteria. Bacteria produce differently than we do, so their reproduction is going to be a cloning process we refer to as binary fission. If we imagine the nucleoid here in red, we need to copy that, create some additional cytoplasm, and one individual, which was kind of a rod-like shape, can then move nucleoid to each side of the cell and pinch it off in the middle. So this seems a lot like mitosis, how we may have normal cells within our body grow and then divide after getting rid of the nucleus and copying the genetic material and reforming a nucleus in each new daughter cell. The fundamental difference with binary fission, of course, is that there's no nucleus involved. This is a somewhat simpler process. One thing we're not maybe addressing here, which we can't see in this picture, is that in addition to this nucleoid that must be copied for this binary fission process, we do have that other piece of DNA called the plasmid. And that the plasmid, which is a small circular piece of DNA, can be transferred through something called conjugation, or sometimes referred to as lateral transfer. And that's significant to us because that means that one bacterium can actually send a copy of maybe a gene for antibiotic resistance to a neighboring bacterium in the kind of equivalent of sex, although it really isn't a full exchange of DNA like sexual reproduction might be. So in conjugation, we're going to get passing of DNA, could be a plasmid, could be something else, And it's going to go from one cell to another. So we see here a plasmid in the cell on the left. And it has what's called a sex pilus here, which is going to be able to connect to an adjacent bacterium which is lacking the plasmid. And so we're going to be able to have this copy of a plasmid work its way over into the bacterium that lacks that, so that at the end of this, both of these cells have whatever DNA is on that plasmid, meaning that this new cell has picked up some new capabilities as a result of receiving a copy of the plasmid from the other bacterium. Now, plasmids can also be picked up. Let's say if a bacterium is destroyed, another bacterium in that area may be able to pick that up as well, and then to be able to produce proteins that are basically included in the instructions of that DNA. So if we want to think about how bacteria are going to cause disease, of course that's going to happen in a lot of different ways, but it kind of comes down to this idea that the bacteria can grow. Let's say we could get a large number of bacteria growing, dividing, increasing in population within our intestines, and that could cause us some illness if we happen to have intestinal problems associated with the bacteria. Or bacteria can produce toxins. So, for example, the bacterium which causes tetanus produces a toxin which is going to cause things like lockjaw, that's a stiffening of the jaw so that can't be moved, and overall body paralysis. So it's going to affect essentially how motor neurons are going to work. They're going to become hyperactive and engage muscles to cause them to maintain these contractions. You probably know tetanus as something you get tetanus shots for, and we often think about stepping on something. So stepping on a rusty nail or something like that might introduce this bacterium into our system. So of course we want to make sure that we have our vaccines up to date to protect us from this kind of condition. Now MRSA is something a little bit different and a little bit interesting. MRSA is methicillin-resistant Staphylococcus aureus. So we see Staphylococcus aureus here. It's not a very interesting looking thing, but then again we can say that for bacteria that they often have some basic shapes, spheres, rods, or what we call spirochetes, which are kind of a little twisted thing. So they may not look that exciting from the outside, but we want to think about what these things are doing. This MRSA is resistant to many common antibiotics, the psyllin group we've talked about. So penicillin, amoxicillin, those kind of things will not be able to fight MRSA. And we see a MRSA infection down here as a skin infection. And of course, some people might confuse this with a spider bite. So people often think that they get spider bites. They get fewer than they really imagine. But MRSA here is actually going to be much more serious in that this can potentially spread to other areas and we may have some trouble controlling it. So this kind of skin infection is common among athletes that are sharing equipment. And so we might often see maybe a MRSA outbreak in a gym, much like we might see a MRSA outbreak in a hospital. Now the good news is that through kind of new protocols which help to make sure that we have a increased cleanliness within hospitals, MRSA rates have dropped in many hospitals over what they have been in the past. All right, that's going to take us into viruses. Viruses are non-living. They are acellular, and of course we recognize that anything that's going to be classified as living has to at least be a cell. Not only that, but viruses are obligate parasites. That means that they have an entirely parasitic mode of life. They have to use other living cells to replicate because they're too simple. Not only are they small, but they basically consist of DNA with an outer coating of protein. So this is called the capsid on the outside, but the inner core is going to be DNA in this case, or in the case of some viruses, not even DNA, but rather RNA. But there still will be a nucleic acid in there carrying genetic information. So simple, non-living, and because they don't have mitochondria, because they don't have endoplasmic reticulum, because they don't have ribosomes, because they don't have a Golgi complex, they need to utilize the machinery of another cell in order to replicate themselves. So how do they do this? Well, here we can see the attachment of a virus to a cell, and it's going to basically get inside of the cell and introduce its genetic material. So here's some viral RNA. It uses an enzyme called reverse transcriptase if it is indeed an RNA virus, this would be kind of the thing that HIV might do, to basically create a section of DNA which is going to be viral DNA, and it introduces that into the host DNA. So now the host can actually go ahead and create some RNA, send it out to ribosomes, which are going to create the proteins which are essential for the assembly of the capsid of the virus, and copies of viral RNA that can be put into those capsids for export. Now the budding that we see here is basically going to be this cell releasing new viruses, which of course, since you're ill, you may be spreading in some way. Maybe it spreads from your blood, maybe it's going to spread as you cough. Those kind of things can transfer this to someone else so it can infect them. All right, one such disease that's caused by a virus would be influenza or the flu. And so this affects 5 to 20% of Americans annually. Obviously, we have bad flu years, we have better flu years, and we have a lot of symptoms that may occur during the flu. Of course, runny nose and cough may be associated with the spread of this. We may be more concerned about things like the fever, the head and body aches, and potentially nausea and vomiting, although a lot of people won't get that with each flu. We tend to get that from other stomach bugs, which are going to be different viruses for the most part. So one big question is, why can I get the flu each year? If I get the flu, and if I fight it off, I should have maybe some memory cells which are going to be able to recognize this and cause me to not get it again. But it ends up that that doesn't work that way because the flu tends to mutate pretty quickly. And one reason for that is because this is going to contain RNA. So when we have RNA containing what we call retroviruses, we tend to get relatively rapid mutation. And this is one of the things that can cause us to get vaccinated for the flu and still end up getting the flu, because when they create vaccines, they need to figure out what kind of flus are out there and try to figure out which kind of flus are going to make their way to the United States. And sometimes they're right and sometimes they're wrong, which means it's possible for us to be vaccinated and protected from certain strains of flu, and yet still get the flu because new strains are available. All right, so what's bird flu? Bird flu sounds like flu that birds would have, and indeed it is, because other animals also get influenza. And we think about this number, H5N1. So we have different numbered lettered strains of influenza virus. This particular subtype is common in birds and can infect people. Now, it requires close contact, so the kind of people who are going to get the bird flu are people who generally are working with poultry. They have very close proximity to birds and lots of exposure. Now, the issue is once it gets into people, it can then potentially be passed on to other people. Now, for the most part, this doesn't happen that easily because this tends to infect lower in our respiratory tract than other forms of flu, and so it's less likely to be passed on. But we do have some concerns about possibilities for mutation or the combination of the H5N1 with some human forms of the virus, which might make it easier to spread amongst people. And so that's what this picture over here shows. We've got here this particular form of the bird flu, and what we're suggesting is that mutations may cause changes in these spikes that are on the outside, which help it to infect the cells of its target organism. And so with mutations to the spikes on the outside, we could go from something that affects birds easily and doesn't infect humans very well to something that may be able to infect humans much more easily and be passed more easily from human to human. Now, the other thing that could happen is that somebody who is sick with a human flu virus could also get sick with the bird flu virus. And because both of these are infecting their host cells and entering their DNA, we could get something that becomes a combination of those and is easily able to infect humans, but carries some of the genes associated with the bird flu virus. Now, one of the big concerns that we have had about this in the past is that some forms of bird flu have been deadly. That is more deadly than a normal flu, although the normal flu does kill some patients, particularly elderly or those with weakened immune systems. So when we've had a very virulent, a very dangerous kind of bird flu, we've been happy so far that those haven't spread easily amongst people. All right, this takes us to prions, and prions are even more different. So we had bacteria, which are living organisms, but they're prokaryotic organisms, so they're smaller and maybe a little bit more simple than us. Then we had viruses, which aren't even alive, but they do consist both of proteins on the outside and some kind of nucleic acid genetic material on the inside. Prions, though, are just protein particles. So they are infectious. They cause a degenerative nerve disease, referred to in humans as Creutzfeldt-Jakob disease, and we may know this as mad cow disease in cows or scrofy in sheep. In any case, what happens is that we get a shrinking of brain tissue and an increase of space within what normally is a relatively tight tissue, so this is referred to as showing spongiform pathology. Pathology basically is just the signs of illness that we're going to see within a particular tissue, and spongiform obviously is making the brain somewhat more spongy. And that's the common thing that we talk about relating mad cow, scrofy, and Creutzfeldt-Jakob, is that these are going to be some kind of spongiform encephalopathy, meaning that it's affecting the brain. So shrinking brain, and then we get basically an inability to control the kind of motor output. You may see cows stumbling around if you've seen videos of cows with mad cow disease. So what exactly is going on? Well, we think that this is transmitted by ingesting, by eating, infected brain and nervous tissue. So this is one reason why we might steer clear of eating brains of some organisms, and certainly of our own species. We run the risk of spreading these kinds of diseases that spread within the nervous system. And so we have normal prions that exist inside the body and have basic jobs to do related to maintenance of the nervous system. But we have a misfolded disease-causing prion. And what happens is when these things interact, the misfolded, what we call rogue prion, converts the other prions into a rogue kind of form. So we get an increasing number of these misfolded and misfunctioning prions. So we don't get the kind of job done that we are supposed to. We get a loss of nervous tissue, so that's the degenerative aspect of it. We have less and less nervous tissue as this goes by, and plaques occur which are going to prevent the proper function of the nervous tissue, and therefore we have that loss of motor control